Hello and welcome to another Piper Pod. Today we're going to be looking at the peripheral nervous system. Now a lot of the stuff we've covered in the past is going to obviously link into the peripheral nervous system, and the next lesson we'll be looking at central nervous system itself. So starting off pretty simply, peripheral nervous system refers to anything that is not the brain and the spine itself. So the central nervous system is quite small and simple in structure, so it's the entire brain, spinal cord, and the tiny little bit of extension from the spine itself, which is where the neural cell will start in a lot of the systems. So the function of the peripheral nervous system is all the other nerves that we have outside of it, that branch out from the cranium and from the spine itself. So its main function can be broken up into three things. It has a sensory responsibility, and that's monitoring everything to do with the environment, both internal and external. It also works hand in hand with the integrated senses. Okay, so it allows the ability to transfer information to the integrated senses in order to be best utilized to become a motor output. Okay, so obviously coordination of all our voluntary and all of our involuntary responses based on the interpretation of sensory input. So that's the whole function of the peripheral nervous system, obviously linking in with our central nervous system. So a neuron, when I say the word neuron, it simply means a nerve cell. Now we'll have a little bit of a closer look at nerve cells later on, but a neuron of the peripheral nervous system can be broken up into really two, but there's actually three different functions. Okay, so we have our sensory, our afferent neurons, and they're responsible for carrying things back towards the central nervous system. Okay, so they are all about our sensory receptors, and they send things from the organ back to the central nervous system. Okay, the other neurons we'll find will be motor or efferent neurons, and they're about carrying impulses away from the central nervous system in order to create an effect. Okay, so efferent, effects, that's basically a good way to remember it. And then there's a few little neurons in between called your interneurons. Okay, and they're the ones that essentially make things happen. Okay, so it's not as simple as message coming in, message coming out. We need another neuron involved in sending it to a different area for interpretation, maintaining homeostasis within the body, supporting us in our arc reflex. Okay, all these things are taken care of by an interneuron, and they are mostly found in the spinal cord themselves. Okay, we'll talk a little bit more about that as we travel along. So looking at a typical neuron, okay, it's pretty hard to define, but this is essentially what you'll find. There's two specialized processes, okay, which are done in their own version of cytoplasm. Okay, so their own version of cytoplasm where activity is done. First one is your dendrites, and there is a huge outlay of root system or branches, if you will, and they're responsible for bringing signals to the cell body before it can be transferred along the axon. Okay, so a lot of the dendrite side of the house for the motor neurons will be found within the central nervous system. Okay, your dendritic side of the house, most of them will more or less be found within the central nervous system, because it's not to be mistaken with our receptors. Okay, so our specialized neural structures of receptors, it's not necessarily dendrites that do that. Okay, the dendrites are the ones that start the process from the central or interpret it in the actual central nervous system itself. From there, the other function is traveling or sending that information that's collected by the dendrites down the axon. Now, different neurons will be shaped or protected different ways that we'll look on, but they all will travel down from the actual dendritic area through the axon until they reach what's known as the axon terminal, where a process will start. So that's the way, in a nutshell, neurons work. Now, certain neurons within the actual nervous system themselves will be covered with an insulation layer. So these insulation layers are high lipid based protective sheaths called myelin, and they're responsible for being those conductive insulators. So they speed up the signal that gets generated through the dendrites, through the axon. So a neuron that is insulated with myelin can transfer things quicker. Now we'll look closer at the two different structures of the neuron, both myelinated and non-myelinated ones. But at this stage, just know that myelin sheaths exist around the neuron. Their job is to aid in conduction. And depending on what nervous system we're dealing with, it's produced by specialized cells called Schwann cells for the peripheral nervous system, or oligodendrocytes for the central nervous system. So both of these specialized cells are responsible in the production of the myelin sheath. So they'll come along, they'll encase the axon, and then they'll just start building layers upon layers upon layers of this myelin sheath, increasing the insulation of that conductive material that we'll talk about later on. Okay, so that's what the myelin sheath does, and that's the cellular structures that form it. Okay, so from there we're going to look at the breakdown. Now, as far as pure structure goes, not including the brain, the peripheral nervous system has a lot of branches. As we know, it is made up of the cranial nerves and the spinal nerves, but from there they have different branches that can do different things. So we've already talked about your sensory afferent division, which is obviously responsible in sending information towards the central nervous system. It is the peripheral nervous system, in order to be interpreted for some sort of effect. Now we have further subdivisions within the sensory side of the house, which looks at our somatic. Best way to describe it is our external environment. What we can touch, what we can sense, hear, smell, all that sort of stuff comes from your somatic side of the house, okay? Your visceral side of the house is all about your internal organs, okay? So this is where we start looking at chemoreceptors, specialized follicular baroreceptors within the organs themselves, and they will send information as well towards the central nervous system in order to stimulate some form of an output from the motor or the efferent division. Okay, so that's what they do. So as far as your special senses go, taste is involved in visceral. Sensory, it's kind of easy to remember because it's an internal structure, your tongue is. Okay, so that's the two different types of sensory division, somatic versus visceral. Either way, they're all involved in interpreting information, bringing it towards the central nervous system for analysis, which will send it back out to your motor division. Now your motor division is really broken up into somatic, so we can look at somatic and brachial as pretty much the same thing. The only reason there's a division is because somatic spinal nerves, brachial motor, we're looking at our cranial nerves. That's why there's that slight division within there, but it's all about our input with skeletal muscles. Okay, so our somatic motor has got to do with skeletal muscle control. Okay, so it's not always voluntary. We can't just say voluntary and involuntary side of the house, even though it's mostly correct, because there's certain aspects within the somatic motor side of the house that is done on an involuntary basis as well. Okay, but most of the time, everything to do with skeletal muscle is on the somatic side of the house, and your visceral motor side of the house is all the activation of non-skeletal muscle. So your smooth muscle, your cardiac muscles, and your glands. Okay, so this is what affects your organs. Now your visceral motor is broken up into two more subdivisions, okay, and that obviously being the parasympathetic and the sympathetic division. So all of these things work together in order to maintain homeostasis. So you will find they work hand in hand with the endocrine system, okay, cardiovascular system, in order to relay an output, in order to help maintain our homeostasis within our body. Starting right at the top of the head, we'll look at our cranial nerves. Now, the reason they're called cranial nerves is because they start in the cranium and then they'll branch out mostly to the face and neck. Okay, they'll pretty much mostly just hang around the face and neck, except for your vagus nerve, which can travel down to the rest of your organs. Two of them, the first two in fact, will come out of the actual frontal aspect of the brain, where the rest of them will branch off from the pons or the midbrain. Now, there's an entire podcast on the cranial nerves, side of the house. I'm not going to go through each one or its function, because if you want an in-depth knowledge of that, you're going to go back to that one. But what I will mention in this stage is, because we've mentioned our motor versus sensory side of the house, is that cranial nerves are broken up. Okay, they can be either sensory or motor, or they can be both. Now, there's only three sensory themselves. Okay, and that's olfactory, optic, and vestibulocochlear. Okay, so smelling, seeing, and hearing. Okay, even though equilibrium is in there as well, it's still your sensory side of the house. Now, most of the other ones associated just with muscle, so movement, can be considered your motor ones, just your motor ones. Okay, but when you have a special nerve, like your trigeminal, for example, now it's obviously associated with the sensation of the side of your face, but it also has motor responsibility to the jaw. Glossopharyngeal, another one, movements of your pharynx, but it's also got to do with your gag reflex as well. Okay, so that is both. Now, these mnemonics, now everyone has learnt their own sort of mnemonic for the cranial nerves themselves, so I'm not going to go through that one. Now, there's two mnemonics to determine whether or not they are sensory, motor, or both. Some say marry money, but my brother says big boobs matter most. Okay, so if you write that down from 1 to 12 and correlate it with the 1 to 12 of the actual cranial nerves, you'll have a nice easy way to remember which ones are it, which is helpful when you're taking a test. What I personally like to do is think about the function of the cranial nerves, which can pretty much help you determine whether or not it is sensory, motor, or has both of those functions. Okay, so whichever way you like to, have a look at it, and like I said, go back to that original podcast on the cranial nerves to get a better idea. From there, we have our spinal nerves, and they branch out from the spine. No big surprise there. Now, they're grouped in a whole bunch of different areas, and it's associated with where on the vertebrae they come out. Now, when you hear the word plexus, it means a gathering of two or more nerves. Okay, so the main plexus within the actual body is your cervical, brachial, then you have your lumbar and your sacral plexus. Okay, so from there, those regions, you have a large cluster of nerves that will come out together. But when we're dealing with spinal nerves themselves, they're like the cranial nerve, they're pairs, so we're not dealing with one. Every time we mention one level, we have two. Every time we mention one cranial nerve, there's in fact two. And they branch out from the vertebrae. Now, the reason it goes all the way to C8, when you know full well there's only 7 vertebrae in the cervical region, is because above the first one, the first nerve comes out. Then obviously you've got an extra one all the way down. So there's 8 nerves that come out of the cervical, 12 of the thoracic, nice and easy to remember, 5 for the lumbar and the sacral, and then there's 1 that comes out of the cochlea, okay, the actual coccyx itself. So the coccyx has its own one little nerve that comes out of it as well. Okay, so obviously all that added up gives us 31 pairs of spinal nerves. Now, unlike your cranial nerve, every single spinal nerve has both a sensory and a motor output. Okay, so we have 2 different branches that will come off from the spine or the central nervous system, and we have what's known as a dorsal root and a ventral root. Now, dorsal root will hold your sensory side of the house initially, and your ventral root will hold your motor side of the house. Okay, but they will swap, and each one of those will go to the areas of the brain. So each one side of the body will have both a sensory and a motor nerve going to that region. Okay, so there's not a division where obviously half the motor goes one way, half the sensory go the other. It will cross in the middle, and they'll have access to both sides of the body. Now, looking at this picture, we'll see that the sensory side of the house has what's known as ganglions on them. Okay, now a ganglion is a way of saying a collection of nerve cells together. Now, the reason they are there is because they're there for initial sensory interpretation. Okay, so they're there. They're not on the motor side, which we'll find out why that is But on the sensory side of the house, it's needed in order to help relay messages to a certain area. Okay, that's why you'll find ganglions on the sensory side, but not on the motor side. Okay, and we'll delve a little bit more into that when we look at the specific nerves themselves. Okay, so looking at our sensory side of the house, what our sensory does is it picks up stimulus. Okay, and the way it picks up stimulus is with specialized organs around the body. Now, two different types of receptors really exist. So you have your exteroceptors, which is involved in gathering information from the exterior. Is it hot? Is it cold? Is there vibration? Did that hurt me? Did it not? Things like that. Okay, and then you have your interoceptors, which are all about receiving information from the organs, so your internal side of the house. Now, a receptor is designed to pick up your fine changes in a whole bunch of different things. Okay, you've got chemicals, you've got movement, you've got pressure, you've got pain, even electromagnetic fields sometimes. Okay, depending on, there's a lot more in other species, a little bit in humans. But the point is we have specialized receptors all over our body that are designed in picking up different sorts of stimulus. Okay, so your mechanoreceptors are all associated with movement. Chemoreceptors pick up changes in chemicals. Electromagnetic obviously does that. Thermoreceptors are changes in body temperatures, often found in our skin. Baroreceptors are found in our hollow organs in order to denote pressure. Proprioception helps us basically maintain an erect position. Right, walking, running, basically tells us where we are in time and space. And a pain receptor helps us from increasing damage. So it picks it up early. And a lot of these are associated with vibration as well, which is another sort of receptors that you can find. And each one of them will interpret changes in the environment and send that change to the central nervous system to be analysed before it can come back with some form of motor response. And that's what the receptors will do. That's part of our sensory nervous system. Okay. Now, the way that we classify that clinically is by the use of dermatomes. So each one of our spinal nerves, as it branches off, and some of our cranial nerves for the face, will be associated with a different region of the body. Now, the way we define that, it was the term dermatomes. So the map on the left is pretty much a classic dermatome map that we've all seen. We sort of get a little bit confused of how it all works because we're looking at the anatomical position. Now, if we look at the one on the right, we've laid the spine out and we've got the spinal nerves draping down as they would if we were to lie the spine along someone's back. Now, from there, it sort of gives us a more easy understanding of where the different regions of the spinal nerves are associated with determining or basically taking care of those particular receptor zones. Okay, that's what your dermatomes will do. Now, obviously it can be used for clinical reasons, but it's also just an understanding of how the body will communicate with itself in order to take care of different regions of the body, okay, and what spinal nerve is responsible for which one. Okay, so dermatomes are associated with mostly the sensory side of the house, and we'll learn a little bit more about different nervous systems and how nerves will travel to different organs, and that will give us a better understanding of things like referred pain and things like that. Okay, but just wanted to show you that there's an alternative to the old dermatome map, which I think is a lot more user-friendly. From there, let's have a look at nerve structures themselves. Okay, so we have two systems that we spoke about. We have the somatic and we have the autonomic nervous system. Now, the autonomic nervous system is obviously the nervous system associated with the parasympathetic and the sympathetic nervous system that we spoke about. The somatic nervous system is smooth muscles. Now, we've already spoken about that when we looked at the muscle physiology itself, but just if we look at the bottom with the somatic nervous system, because it's easy, we can see that there is one neuron associated with the somatic nervous system. So it goes straight from the spinal cord. The motor neuron is just one. So straight from the spinal cord, straight to the actual muscle that it wants to activate. Now, the autonomic nervous system is a mixture of two different neurons. Okay, so we have what's known as a pre-ganglionic neuron and a post-ganglionic neuron. Now, depending on the actual nervous system that we're dealing with, so parasympathetic, sympathetic, things like that, will determine what sort of neurotransmitter will actually be utilized. But at this point, I just wanted to highlight that the autonomic nervous system, each pathway, is made up of two different neurons, whereas somatic nervous system is made up of just the one. Also, the somatic nervous system neuron is myelinated, completely myelinated, whereas the autonomic nervous system, just the preganglionic neuron, is myelinated. And we'll talk more about that as we go along. Okay, so when we look at our pre and post ganglionic neuron for the autonomic nervous system, if we focus purely on the preganglionic, this is the side of the house where the cell body sits in our central nervous system, as I spoke about. And it has, its axon is myelinated, and it will extend out until it finds an autonomic ganglion. Okay, now the reason that this happens is because then the one single nerve can affect different routes of a particular ganglion. Unlike the somatic side of the house, where we're interested in just passing on information from that to that muscle, no problems at all, maybe a branch off. This one here has the ability to transmit different chemicals to the actual ganglion in order to elicit different responses across the board. Okay, and also different responses associated with sympathetic or parasympathetic. The postganglionic neuron, okay, its cell body, it obviously is nowhere near the central nervous system. It's right within the peripheral nervous system, sitting with that ganglion itself. So a cluster of cell bodies will be found within that ganglion. Now, its axon is not myelinated. So that fiber from that ganglion to the actual effective area is not myelinated, which means the impulses are transported a little bit differently. And we'll discuss reasons why that can be advantageous in a lot of cases and what the body has done to essentially accommodate for that. So having a closer look, we need to look at what the autonomic nervous system does. So obviously we have a breakup of the sympathetic and the parasympathetic nervous system. The sympathetic, as far as division goes, is what's known as the thoracolumbar division. So its pre-ganglionic cells are found in the thoracic and the top aspect of the lumbar. So number 1 and 2 aspect of the lumbar around the spinal cord. So the entire sympathetic nervous system division is in the middle of the spine. The entire thoracic and the lumbar takes care of our entire sympathetic nervous system. The parasympathetic is extremely unique. Okay, so that's what's known as a cranial sacral division. Okay, so its pre-ganglionic cells are found in the brain itself as part of the cranial nerve. Okay, so 4 of the cranial nerves are associated with the parasympathetic nervous system, and the other part of the parasympathetic nervous system is found all the way down in the sacrum itself. So looking at them a little bit closer, we'll look at the sympathetic division. Now obviously, I'm not going to insult your intelligence. You all know that the sympathetic nervous system is responsible for what can only be described as our fight or flight response. Okay, so anything that's emergency based, anything that elicits an emergent or survival response is taken care of by the sympathetic nervous system. Okay, so the sympathetic nervous system is associated with speeding up certain functions and turning off non-essential functions itself. So it will speed up your heart rate, it will dilate your pupils, it will increase the bronchial dilation of the lungs, it will release sugar, it will release fats, it will slow down digestion during those times, and that's what it does. It is all about survival. Now, when we look at the division of the preganglionic versus the ganglionic neurons, okay, we see that the preganglionic neurons are relatively short. Okay, so they're very short neurons, and they have a loop that assists for the lower half of the body. So from L2 down, there is a sympathetic chain ganglia that will help get neurons extend the neurons to the lower part of the organs. Okay, now the gangliotic nerves are very long, and they stretch to the organs from the actual preganglionic neuron that is found. Now, Harry mentioned that the autonomic nervous system has those two different cells. Okay, so we have that side of the house in order to assist in neurotransmitter cooperation, realistically. Now, I want to draw your attention to the adrenal medulla. So if you look at the actual adrenal medulla and follow it back to the actual spine itself, you will see that that is just a preganglionic neuron. Okay, so that itself actually is just one neuron. Okay, so it's one of those unique neurons associated with the autonomic nervous system that is just one special neuron that will transfer straight from the spine to the adrenal medulla, obviously stimulating the release of adrenaline, noradrenaline, which is associated with assistance within the sympathetic nervous system itself. Okay, so that's essentially why we have that division. Okay, so that's the sympathetic nervous system itself and the division. When we look at the parasympathetic nervous system, we see that things are slightly different. And by slightly different, I mean exact opposites. Now, by rights as well. Okay, so the parasympathetic nervous system best described as the exact opposite to the sympathetic nervous system. So everything the sympathetic nervous system does, the parasympathetic does the opposite. Okay, so it is involved in all of our recovery side of the house. Okay, so resting, removing waste, absorbing nutrients, all that sort of stuff, recovery, metabolism, that's all parasympathetic nervous system. Now, as you can see, most of the functions of the parasympathetic nervous system are controlled by the actual cranial nerves themselves. Okay, so cranial nerve 3, 7, 9, and 10 are associated with the actual parasympathetic nervous system because they extend out and assist in a lot of those functions. Now, the vagus nerve obviously is one of the most important aspects of the parasympathetic nervous system, especially when we're dealing with medical emergencies. Okay. Now, the lower half of the body is controlled by the actual spinal nerves from the sacral region, but it's also assisted by the branches of the vagus nerve itself. Okay, so they work hand in hand together. Now, if we look at the preganglionic versus the ganglionic neurons, you will see that there is the parasympathetic nervous system has enormous preganglionic neurons and then a very small ganglionic neuron or postganglionic neuron from there. Okay, so the pre will travel all the way to the organ itself and then it will meet its ganglion and then it will essentially move on from there. Okay, so that's a big difference structurally between the sympathetic and the parasympathetic nervous system itself. Okay, so a lot of the time the parasympathetic nervous system is involved in the inhibitory of the sympathetic nervous system. So if things get out of hand and the body needs to rest, the parasympathetic nervous system needs to be able to get to the source directly in order to have that inhibitory response with its neurotransmitter. Okay, so that's what the parasympathetic nervous system looks like, and there's some of its functions. We're just going to have a quick look at how the actual neural impulse is passed on. Okay, so we looked at the anatomy, we looked at the breakdown. Now we're going to look at how the nerve actually has that impulse and how it passes on that information and how that information is further relayed, especially when we're dealing with the sympathetic and parasympathetic, where we have that ganglion junction associated with our mix. A nerve impulse is generated by a change in the electrical charge that's found at the membrane. Okay, so each membrane has what's known as an optimal voltage that can elicit a response, and that's what's called a membrane potential. Now, the way a membrane potential starts is a neuron at rest, or most things at rest, has what's known as a resting potential. So this is the potential charge, okay, so the actual charge that's found within the neuron when it's not stimulated by any external source. Now the reason that we have a low millivolts is because of the associated structure that's found inside the cell compared to outside the cell. Okay, so there's a lot of negative elements that is found inside the cell. Okay, now I know when we first started learning this, we learned potassium and sodium, and that was the only thing we needed to know. And potassium is slightly more negative than sodium, therefore that's what causes it. And that's only partial truth. Okay, absolute partial truth. The main reason that the inside of the axon is so negative is because of its large proteins that are found inside that have a negative charge. So our weak acids, our large proteins, they have a negative aspect to it, and they're too big to get out of the cell. As opposed to outside, in the actual interstitial fluid, where there's no proteins. Why would proteins be floating around the interstitial fluids? Okay, you'll find the odd one, but you wouldn't find many. They're not useful there. So they'll be pushed into the bloodstream, pushed into cells in order to be utilized from there. Okay, so that's the main reason why we have that negative inside versus the positive outside. Now, obviously, potassium plays a role, obviously, and so does our chloride on the inside. On the outside of the cell, the main player in the whole charge is obviously our sodium, and chloride essentially helps out a little bit as well. Now, when a neural impulse comes through, okay, so think of it as every neuron, we've already mentioned that it travels one way. Okay, now the reason that neurons can travel one way is because of its cellular structure. So it starts from the dendrites, and then it has what's known as a target area. And the target area is where the initial impulse will start and then travel along the axon in one direction. Okay, so as the neural impulse comes in, a few things will happen at the start. First of all, chloride will just start leaking out of itself. That will have minimal effect on the actual charge of the membrane itself. Most of the time, the charge comes from our actual stimulus, our neurotransmitters, things like that. As it comes along, that increase in voltage, most of the time it gets to around -55, -60, and that is enough to open our voltage-gated sodium channels. Once one of them opens, sodium just flushes in, absolutely flushes in to the inside of the cell because the sodium channels are open. Now that increases the membrane charge up to a point of around about +35. Okay, once it gets to +35, your potassium channels open up, and that flows out our potassium the other way, lowering that membrane potential as well. So therefore, it brings it back down. Now, the way that helps with an axon is because once the membrane meets a certain threshold, okay, it's what's known as an action potential. Okay, so an action potential is what's known as an all or nothing system. Once we reach that -55 and the sodium channels open, then obviously sodium comes in, increasing that charge. The same amount of sodium will come in, we get the same amount of charge each time. As the charge increases, the sodium channel next to it is obviously charged and opens up, bringing sodium in, and so on and so forth, all the way down the axon until we get to the axon terminals itself. Okay, so that's what happens. So when a neural impulse occurs, it's not one action potential, it is multiple action potentials right along the axon itself, opening sodium channels all the way, and then relaxing as it comes through by opening potassium channels as they go down. Now what's interesting enough is when we look at how do we tell the difference between a feather hitting our shoulder compared to a brick hitting our shoulder. If the action potential is all or nothing, as soon as I open one of those sodium channels, then I'm going to get the exact same response every time, regardless of what opened it, how much stimulus opened it, it's still going to be the exact same response. So the big question is how do we feel the difference? If all of our action potentials are the same, technically everything should feel the same. Now the answer is not a question of voltage, but the question of how many subsequent stimulus come through the dendrite itself. So if a feather hits you, you will get a stimulus that's quite small, and that will pass on the action potential, and it'll be, if only a few stimulus will pass through, meaning only a few action potentials will go along the axon within a particular time frame. If a brick hits your shoulder, then you will have subsequent stimulus hitting that target zone, which will respond in multiple action potentials and multiple neurotransmitter release for a bigger, more exaggerated response. So whilst the voltage of the action potential is always going to be the same, by increasing the number of stimulus that comes in, that's how the body can distinguish between the difference in problems or the difference in the stimulus itself. So that's how the neural impulse works, all the way up to the axon terminal itself. Once it gets there, so once it all passed through, we'll just quickly mention something known as the sodium-potassium exchange, because obviously now the body has to re-regulate itself. Now, some sodium will leak back out of most of the neurons just naturally. Some potassium will come in just naturally, but the majority of the actual exchange is done by the sodium-potassium exchange pump, which takes energy. So that's where it will bind 3 sodiums, use energy in order to push it out of the cell, active transport, against the volts. Okay, this uses what's known as visceral transport. And at the same time, it will draw in 2 potassium into the cell, burning up that ATP. So this process here is actually what's responsible for most of our ATP use, but we do not have to wait for every single sodium potassium to swap, because all we're measuring at the moment is the actual influence on the membrane itself. Okay, it's not taking into account anything behind us within the cell, which realistically is full of potassium. Okay, and it's not taking into account anything within the actual interstitial, which is full of sodium. Okay, it's sort of like having an entire room and only accounting for changes that happen at the actual wall paint itself. Okay, so whilst this happens, the body does not have to wait for every sodium potassium that was moved to be moved back before an actual potential can be started again, because in reality we're dealing with small percentages of these electrons each time. Okay, by electrons I mean electrolytes. Now, things that assist in this whole process. Now, we talked about the difference between myelinated and demyelinated cellular structures. So the myelinated cellular structure is obviously associated with high speed conduction, because what happens is as the impulse comes down, there's no need to open any other gates, any other channels, until we get to the junctions of the myelin sheath themselves. Okay, and they're called the islets of Auger. Now, the cool thing about myelination is when we're born, not every one of our neurons are myelinated. So as we develop and go through time, then we start myelinating our neurons. And what happens is the neurons that look like A will eventually end up looking like B. So the sodium and potassium channels, voltage-gated sodium potassium channels, will actually migrate from the axon to the islets of Auger. And from there we'll have a highly compacted area within the axon that has these voltage-gated sodium and potassium channels. Now, the big research side of the house is when we're dealing things with like multiple sclerosis, which is associated with demyelination. The only reason that's a problem is because there's no voltage gate there anymore. So the impulse is lost. It can't be conducted anymore because there's holes in the myelin sheath. So the impulse just dissipates like energy does, but there's no sodium gate there. Okay, so when we're developing, our sodium channels can move, but for some reason, and we're still researching it, we don't know why the body can't adapt and move them back to areas of demyelination. Okay, so that's just an interesting thing to think about as we go along. Now, the ones on the left, they have to work like a wave effect. So the only way the impulse can be carried on is if all those sodium channels are open, all the potassium channels are open as the wave effect goes along. It's slower, but it allows for a delay in stimulus and usually a cooperation in stimulus as well. Now, once we get to that axon terminal, we obviously have what's known as the synaptic areas. Now, each neuron has thousands and thousands of these synaptic knobs at the end of the nerve itself. Okay, so there's so many input synapses at the dendrites and the cell body and the axon terminal. Now, at the synapse, it's responsible in transferring information from the neuron to another area, whether it's a gland, whether it's a special type of muscle, or whether it's another neuron itself. Now, there's two types of synapses, communications that exist. There's an electrical and there's a chemical. Now, the electrical we'll just mention quickly because it has specialized tubules that can transfer electrical current from the presynaptic cleft to the postsynaptic cleft. That's it. And it just passes that channel on and then it continues it along. The major way in which things occur is through chemical synapse. Now, chemical synapse is conducted by neurotransmitters. Now, the body has a series of neurotransmitters, and each one of them are responsible for stimulating a different receptor site. Okay, so neurotransmitters come in, they're released by the synapse, they travel through the synaptic cleft, and they bind to the postsynaptic cell, which causes ion channels to open. Depending on which neurotransmitter comes in and which receptor site links to will determine which of those ion channels will open, and that's what neurotransmitters will do. Now, you can look a hell of a lot into neurotransmitters because a lot of the neurotransmitters can be released from the same synapse. Okay, the same synapse can release a number of neurotransmitters. Now, these neurotransmitters can have a single, a cooperative, an inhibitory, an excitatory, or a second or third response. Okay, so a lot of them are associated with activating C-reactive protein or calcium that will do it, and it gets deliciously complicated from there. So I'll try to keep it as simple as possible, and we'll look at the neurotransmitters that are associated with the actual parasympathetic nervous system directly, okay, not indirectly. Okay. As mentioned, nice and simple, we'll just look at the excitatory synapse and the inhibitory synapse. So if the neurotransmitter is associated with excitatory, then it will open up sodium channels, therefore increasing towards that action potential. If it's inhibitory, then it will open potassium channels, which removes the possibility of having and starting that action potential. Okay, so the different neurotransmitters, different effects. Okay, so when we look at our autonomic nervous system and its neurotransmitters, we have what's known as an andrenic and a cholinergic. The andrenic pathways are associated with the sympathetic nervous system. Now, the preganglionotic neuron that stimulates the ganglion is acetylcholine. Okay, it is always acetylcholine from the preganglionotic neuron. From there, once it stimulates the postganglionotic neuron and travels down, the neurotransmitter released most of the time is norepinephrine. Okay, so that's the neurotransmitters associated with the andrenic side of the house. The cholinergic side of the house is acetylcholine, both ways. Okay, but we have two different receptor sites, which you can read a lot into. Okay, and that is your nicotinic receptors versus your muscarinic receptor sites. So looking at the sympathetic and what the noradrenaline does. So remember, it's associated with our excitatory survival sort of response. Okay, so this stimulates, so it has an excitatory stimulus on the alpha-1 and beta-1 receptors. That increases heart, positive inotropic effects, and your vasoconstriction. It has an inhibitory effect on alpha-2 and beta-2. Now that actually prevents the release of noradrenaline within those actual areas itself. Okay, and in turn focuses on the smooth muscles and aids in your vasoconstriction, blood pressure control as well. It also has an effect on beta-3, which is responsible for the release of fatty acids that can be used in energy. Okay, so release of glucose, release of fatty acids that can be used in energy. Now, when noradrenaline is released and it's sitting in that synaptic cleft, it will sit there until it goes through a couple of options. Now, all neurotransmitters will stay in place until one of three things occur. It will either diffuse, dissipate out of the cleft, therefore no longer be a problem. It will be broken down by basically enzyme activity, which will break it down, or it will be involved in what's known as a presynaptic reuptake. Okay, so we'll actually pick up the neurotransmitter and reuptake it back into the presynaptic side of the house. Now, neuroadrenaline goes through either dissipation, or it's broken down by the monoamide oxidase, or your acetylcholine methyltransferase will break down those particular norepinephrine, so therefore it can no longer elicit a postsynaptic response. Okay, parasympathetic is dealing with your cholinergic neurons. Okay, so they release acetylcholine, and the acetylcholine will respond to two different receptor sites. Okay, your nicotinic receptor sites, which is found dendrites and cell bodies, okay, within the neuromuscular juncture, and your muscarinic, which is found at your basically your parasympathetic response for a particular organ, is what's known as a muscarinic receptor. Now, acetylcholine, as discussed, will stay active until it is broken down by acetylcholine esterase, reuptaked, and then recycled into the presynaptic terminals. Hey, the last thing we're going to talk about, because we've talked about a lot within the parasympathetic nervous system, is this arc reflex, okay, and how that fits into the whole process. Because what we've spoken about the whole time is the stimulus has to come, be analysed by the central nervous system before a motor response can actually be in effect. Now, most of the time that's true, and even when we're dealing with the arc reflex, it's still somewhat true as well. Okay, but obviously the arc reflex allows us to do things a lot quicker. So we'll start just obviously with a stimulus that we have denoted to be so bad that it's going to cause a lot of damage. Okay, that's picked up by the sensory neuron and it goes into the central nervous system of the spine. From there, it does two things. It needs to talk to one of those intraneurons. Okay, so excitatory intraneurons. That intraneuron itself will deliver information to the brain and to the motor neuron. The reason it will do both is because the motor neuron will essentially give us a immediate response, allows us to contract, get away from the situation, but the traveling to the brain allows us to analyze the situation for second, third, ongoing effects. Okay, so there's no much point moving your hand from the tack if you're going to end up putting it into a grinder. Okay, and that's what that message to the brain is essentially allowing us to understand. Now, big question that often comes up is what happens with people with spinal injuries? Do they still have a reflex arc? Not involved in the brain, there's no communication with the brain, will they have a reflex arc? Now, a lot of the time, fortunately, the answer isn't simple. All I can give you is a good, solid maybe, depending on where the spinal injury is and how bad it is. A lot of the times you need to be able to sense a stimulus in order for this action to occur. Okay, if you can't sense it, that's essentially denoted by the brain, then sometimes it doesn't work. Sometimes if I stimulate the right way, okay, so you can lose certain receptor sites but not others, if I stimulate the right way, you can still have this reflex response associated with even with spinal injuries. Okay, so like I said, there's no easy answer for that one, unfortunately, so all I can give you is a good, solid maybe. Okay, so that's the peripheral nervous system in a nutshell. I understand it was quite a long one, a lot to go through. Hopefully you got something out of it. The next lesson will be in the central nervous system. So until next time, take care.